All right, good afternoon, everyone. Welcome to the HM Hanna Lecture. My name is Fred Schumacher. I am the current acting chair of the School of Medicine Lecture Committee. So part of our responsibilities is to look for some of the best scientists across the country, also across the globe. We've got several endowments that we can work with, and then to bring them in, have them give a talk, have them meet with people, trainee lunch earlier today, and then dinner afterwards. So this particular lecture series started back in the early 1900s. It was a gift from a physician to a friend, or in the name of a friend, to help make sure that we got some of the best speakers across the globe to come in, which I think we've done a pretty good job today. So today we're going to have Dr. Pradeep Natarajan give us a talk. I can list a number of things, and so I think it's fair to do so for some of this, right? So he's Director of Preventative Cardiology and the Paul and Phyllis Fireman Endowed Chair in Vascular Medicine at Mass General. He's also the Associate Director of Personalized Medicine at Mass General Brigham, the Associate Professor at the School of Medicine at Harvard, as well as the School of Public Health at Harvard. He's the Associate Member of the Broad Institute of Harvard and MIT, which is some very big scientists there. So, and he's a star there. So obviously we wanted to bring him in. So looking at everything he's published, I would say the way I kind of would summarize it in one sentence is Dr. Natarajan looks at germline and somatic mutations to understand clinical disease, particularly with cardiovascular research. And through this, he's published over 400 publications. He has several grants that are funded through NIH and through foundations. If you look at eReporter, the NIH grants, it's very interesting to see that a number of them are MPI grants, which means he's a very collaborative person who understands how to work with people and take that science to a whole different realm. The other thing is he's an associate editor for JAMA. He's a fellow or a member of several of different groups. I don't want to mention all of those, but the two things I noticed was over the last 3 years, he's won mentoring awards within the Boston area, mentoring either fellows or trainees, which I think is just one of those things that we don't always mention and highlight, but very important to keep the next generation kind of growing within the field. So it's my honor to introduce and to turn it over to Dr. Natarajan.
Thanks, Fred. That was really kind. Okay, yeah, thank you all for coming, and those of you that are logging in. Hopefully this should be fun. It's intended to be a little bit provocative, but really the goal is to expose you to at least some area of science that we're doing that I think has nearer-term clinical impact and clinical translation. These are my disclosures not relevant to this talk. Okay, so this is down the street from where I grew up. My family still lives there. I was actually just there a couple weeks ago celebrating my dad's 80th birthday. But this is the mall that I would go to all the time. I'm showing this because, a lot of people actually today, interestingly, maybe more than most places, ask me why I do the things that I'm doing today. And sometimes also whether it was genetics or whether it was cardiovascular disease. And for me, cardiovascular disease was increasingly apparent, and it seemed to be common. In the South Asian community, it's now recognized that cardiovascular disease is common, but at that time it was not really well appreciated. But it felt like it was something important that was influencing the community, and thought that it was something that I should think about and focus on. Now, for bonus points, actually, does anyone recognize this image? There's a real image. This is the mall, but I will guarantee that, depending on your age, almost all of you guys have seen this mall.
Orange County.
But where have you seen it? Everyone's seen it. Maybe not. This is where Marty McFly first went Back to the Future. This is our claim to fame in that area. Nobody, at least in the 80s when they filmed it, would go to that mall. And so it was good for a movie spot. The other claim to fame in my high school, Fergie from the Black Eyed Peas, was like a couple years ahead of me. But that's what happens when you go to school in LA, I guess. Okay, so yeah, the human genetics part came later. The reason why it came later is that the tools and the technologies are relatively modern as far as their use as research tools. The first draft of the Human Genome Project, that was 2001. I was in college at that time, but nobody was conceiving of using that to study and to understand common disease. Most of my experiences were motivated by exceptional families and exceptional individuals and trying to understand in the lab what might be driving those exceptional phenotypes, largely related to lipids and atherosclerosis. But then I got excited later on in my training when I understood that there were now tools that we can study directly in those individuals, the target humans, to start to understand mechanisms of disease. And this is using naturally occurring human genetic variation to kind of mimic some of the things that you would perturb in a model system, but to look at it, out in nature, again, to really understand disease directly in the individual. And there, I've listed here many different reasons why we would use genetics and what the applications would be. A key opportunity is that when you, in general, at least in humans, relate an exposure and an outcome, like cholesterol, smoking, gray hair with cardiovascular disease, some of those could represent causal relationships, and some of those might represent confounding, that might be correlation, might be other things that are explaining each of those features. And you wouldn't be able to immediately tell just from looking at those biomarkers or those traits and the ultimate clinical outcome. Genetics has the opportunity of having exposures that are fixed at conception and static across the life course. So nothing else is going to influence the allocation of those alleles and genotypes. And if you establish a genotype-phenotype association, that might represent some causal mechanism. There's separate challenges in trying to disentangle what exactly that causal mechanism, but it is a node that suggests that might be actually relevant for human disease. The opportunity that I'll talk about today is what has emerged is now taking the totality of that information to try to predict risk for an individual patient. And there has been increased interest in this, almost surprisingly so. This is a document that we put on behalf of the American Heart Association a few years ago, really summarizing the state of the science because there was an interest from the broader clinical cardiovascular community and the scientific community to kind of understand what the state is and what the opportunity is. Actually, just earlier this year, the European Society of Cardiology, they put out a clinical practice document on how a clinician should start to think about these scores, indicating this general trajectory. And moving one step further, many of you may not know, but just a couple weeks ago, we announced actually a clinical assay within our health system to test individually in the clinic these polygenic risk scores. This is actually an orderable test that any clinician in the US today can actually order this test. It's a PDF that you mail. This is work that came out of my lab, is run by the health system at cost, and I don't have any financial return myself for this. So a lot of it is based on just general interest of what people conceive of might be relevant in their DNA and how that might influence their human health. Over the last 10 to 20 years, there's been a rise in genetic testing, largely driven by the patients that we see, the large populace, people paying for genetic testing through direct-to-consumer genetic testing. And this is fascinating because, like for a lipid panel, which we've long recognized has relevance to cardiovascular disease, you still have to go to your doctor to go get an order for that so that you can get lipids. But for a human genome, for a genome sequence, you can go pay for it yourself and get that information. And the estimate today, based on this sampling, is that 1 in 5 Americans has done one of these direct-to-consumer tests, these at-home genetic tests. Most of the times, the reason that they're doing it is really to understand their lineage, ancestry, understand their familial background, but almost half of them are doing it because they feel like it might convey some health information. What's also changed, again, over the last 10 years, there has been a substantial rise in the generation of genetic data, particularly in context that it connects with health information. These are healthcare-associated biobanks. This is one consortium of biobanks, and the numbers here are actually much lower than what they are today because they are continually growing in size. This is ours today. When this was written, it was 20,000. Now it's 200,000 individuals and 60,000 have been sequenced. And there are many things that one can do with biobanks, in addition to genotype-phenotype associations, is really understand the various different contexts and the various different exposures that might influence the expression of the genotypes themselves, but also them as individual exposures, because there's lots of different reasons why people will get a wide variety of diseases. This is one such example where we did the first genetic description of our biobank in New England. You can see most of these individuals are clustered in Massachusetts, largely within Greater Boston, but does go up and down the East Coast. And if you look at groups of individuals that are genetically similar, you find that genetically similar groups live in different areas and they cluster in different areas. And when we start to look at genotype-phenotype associations, there's an assumption that the genotypes aren't necessarily correlated with these other exposures. But these do represent important other health exposures that we can start to understand other factors that could be correlated with the genetics, but that also might influence the outcomes. But we see cool things, like almost everybody in the field uses the UK Biobank, for example. And in one type of analysis that you can do is look for founder effects, effective population size when you go back in prior generations. You don't see such bottlenecking in the UK Biobank, but in New England, you see bottlenecking. Basically, early signals, these are individuals of British ancestry, where there's an effective reduction in population size that basically maps back to the founding of New England and some of the early founders of the US that you see in the signatures of folks that are in our biobank. Now, genetic testing is obviously done today. It's done for cardiovascular disease, it's done for cancer, and it's largely within the scope of how we practice medicine today, individuals largely with disease, to understand the etiologic basis, but primarily to understand prognosis and actionable changes that you would do differently for individuals that would have a specific genetic driver of their condition. So genetic testing is really focused within that scope. Now, in my world for coronary artery disease, that is focused on familial hypercholesterolemia. This is a condition that afflicts about 1 in 300 individuals, leads to very significantly elevated LDL cholesterol, the so-called bad cholesterol, and about a 3 to 4-fold increased risk for coronary artery disease. The molecular basis of that condition was well described in the 60s, 70s, and 80s, ultimately culminating in a Nobel Prize, and really a group of, again, exceptional individuals, exceptional families that had a clear exposure that was linked to coronary artery disease, and describing that basis, that causal basis, really formed the fundamental understanding of LDL cholesterol and its relationship with coronary artery disease in the population. Now, we do sometimes do genetic testing for individuals who have severe hypercholesterolemia to see if they have FH, but it is controversial. It has been controversial because, again, for folks who have very high cholesterol, in general, there is consensus, at least for the last 50 years, that you would treat them to manage their LDL cholesterol, kind of regardless of whether they have FH or a strong genetic predisposition. What has changed has been these now larger population-based sequencing studies to understand the prognosis in a less biased context. Because there are monogenic mutations that happen in the population in addition to these exceptional families, you would just not know because those who are coming in the clinical context have other reasons why the disease is manifesting. To really understand penetrance, you have to have an unbiased assessment in a broad group of individuals. Now, what these studies have shown is that has shown that for the same cholesterol level, the prognosis is different for coronary artery disease based on whether you have a strong genetic predisposition to hypercholesterolemia, for mild, moderate, and severe, because some individuals with FH do happen to have mild hypercholesterolemia. What's also changed is that, again, over the last 10 years, there's been an emergence of more expensive lipid-lowering medicines, and really to help allocate those who are going to benefit the most from those medicines, we really want to refine who is at particularly high risk for very high cholesterol. Now, there's obviously, unfortunately, there is heterogeneity in how these patients are managed. Some individuals with FH, in reality, are not treated with lipid-lowering medicines, and there are many different challenges of why significant hypercholesterolemia is not appropriately recognized. But when you go back, obviously, those who are treated with lipid-lowering therapy in these retrospective analyses do have lower risk for cardiovascular disease. It's retrospective because it would be unethical to randomize them to placebo. But the effect estimate, the relative risk reduction, is pretty large, down to 0.3. About 70% relative risk reduction. That's large because in the general population, the hazard ratio for statins is about 0.75 and 0.8. Now this may make sense. Those where their principal driver of risk is hypercholesterolemia, they're likely to benefit significantly from lipid-lowering therapy. Okay, we'll come back to that concept. Now, most individuals with FH, most individuals who go on to get coronary artery disease don't have FH. Cardiovascular disease is a very big problem. This that first panel is deaths attributable to cardiovascular disease in the US. And you'll see a couple interesting things. Obviously, that number is high. It's not, that slope is not consistently going up. And this times with an era where there was now an emerging recognition of modifiable risk factors. And all of this kind of came together with key anecdotes. This is, in the 40s, that's when FDR had a stroke, a hemorrhagic stroke in office, and had uncontrolled significant hypertension for at least a decade preceding. Again, there was some recognition of hypertension, but not really that direct link as an important causal factor. President Eisenhower also had a heart attack when he was in office as well, too. And through those things, that led to President Truman signing the National Heart Act that established NHI, which ultimately became NHLBI, that also established the Framingham Heart Study, and started connecting the importance of these modifiable risk factors. And then in the 1960s, the Surgeon General's report on smoking. And all those things, the culmination of those things, had tremendous population health benefit at all times around that time. So the curve is largely flattened, but it hasn't come down. And there's been a lot of concern that really over the last 10 years, it has been rising. Now, cancer has now actually come up. It is a very close second, but this is kind of a dubious distinction to be number one. But here's another curve where the curve is continually going up, and this is costs attributable to cardiovascular disease. And everything is getting more expensive. People are living longer, so continually dealing with cardiovascular disease is obviously a big problem. Now, in the 60s and the 70s, we described modifiable cardiovascular disease. 2025, we're kind of still dealing with this same modifiable risk factors. One might argue we've hit our ceiling with only focusing on the modifiable risk factors. I will argue that most individuals are still suboptimal for modifiable risk factors, and that still does explain why the event rates are still quite high. And this is from the Framingham Heart Study. This is kind of a mathematical exercise of our guideline recommended risk calculator. Since the inception of the contemporary prevention guidelines in the 1980s, what we've effectively done is tried to estimate what the risk is over the short to intermediate term to really identify the highest risk individuals, bless you, for who might merit from either additional testing, but primarily from lipid-lowering therapy. It's about a 10-year risk estimation. Now, what most people actually don't recognize is that in that assessment, and that's been the same in the 1980s, including into 2025, is that most of that prediction is actually driven by age. Most individuals are having events, or at least in the population level, most of those events are happening later in life. So age is the dominant factor. And then all these other modifiable risk factors for prediction actually just move the needle up and down a little bit. And this is a nice example of this. So the kind of the actionable threshold is an absolute risk estimate of 7.5. And if you just kind of go in the calculator for deciles of age, and then you go up and down across a single risk factor, what really determines whether you're going to be near that category is basically what your age is. And there's a narrow window where you can actually discriminate above and below based on that risk factor. So really, these tools are well suited to predicting risk when folks are in middle age, solid middle age, later middle age. But the disease process starts much earlier. And it's really hard to understand how early. This is probably the best study that looks at this. This is actually looking at US soldiers that were killed in foreign wars. These are folks in their 20s, did not die of cardiovascular issues, so died of trauma. And then there were autopsies, basically looking at their coronary arteries. And about one in 9 individuals, 1 in 10 individuals in their 20s, healthy US soldiers, had already developed atherosclerosis. Now, the prevalence of smoking over years has decreased in US soldiers, but that holds across basically all of the data sets, including those who died in Iraq. Obviously, if they had traditional risk factors, there was an enrichment of atherosclerosis that was present in the coronary arteries, but the prevalence of risk factors is pretty low. So the dominant number of individuals that have atherosclerosis at that early age is not explained by premature development of cardiovascular disease risk factors. So we don't have the tools currently today that really help us identify those who are starting disease early, where you have the opportunity to substantially change the disease trajectory early. Now, there's been a long-standing notion that genetics may play a role, and this has been rooted in family history. And this is a study also from the Framingham Heart Study. This led by Don Lloyd-Jones when he was a fellow at MGH. Actually, Don just came back and is now the PI of the overall Framingham Heart Study. And this says, independent of the traditional risk factors, including LDL cholesterol, if you have a parent with premature cardiovascular disease or a sibling with premature cardiovascular disease, your independent likely risk is almost twofold. And that's independent of the traditional risk factors, saying that there's something else, there's some additional information from the family history. So we take family histories, obviously, in the clinic today, including for coronary artery disease with this in mind. But family history is not in any of these 10-year risk calculators that guidelines recommend, because family history is very nuanced. I mean, I do this all the time because in my preventive cardiology practice, it's often younger folks who have a family history. And that's the one signal. That's the main signal, because they're younger. They don't have a high prevalence of traditional cardiovascular disease risk factors. And what we're trying to do is to contextualize that family history directly for that patient. And so we're saying, okay, maybe that uncle smoked and that's why they got cardiovascular disease. Or dad had hypercholesterolemia, but he was sedentary. Maybe that's why he got cardiovascular disease. But you don't really know. You're trying to basically make a guess directly for that patient. And some risk calculators use it, but it's hard to kind of pull in all those pieces of information directly to the patient. So I do agree with what the guidelines say, is that we need to kind of still keep it separately, because the family history itself is not sufficiently quantitative that is accounting for these factors that could be brought in. But based on those studies that Don and others had done that have shown that familial aggregation, empirically suggesting that DNA sequence variation that is common in the population could influence risk for coronary artery disease, that then led to the development of genome-wide association studies. The first one that was done was when I was finishing medical school for coronary artery disease. There were 3 that were published together. And effectively what that is you take a population of individuals or a group of individuals, label that who have coronary artery disease or disease of interest and don't have disease, and then you systematically profile the genome, and you're looking at differences in allele frequencies between the two. And you're seeing what are the alleles, what are the regions of the genome that are enriched for genetic variation in cases relative to controls. And the technology for this has been through array-based technology, where you sample about 1 million base pairs, and that's a small subset of the 3 billion base pairs that are in the human genome. And that's because among the common genetic variation, there is a lot of correlation, and so we can often fill in the gap to about 10 million. But this is really designed to look at genetic variants that are very common. And when I say common, I mean 1% in the population and above. Now this schematic is a little bit older, but what it's intended to represent is that there are a number of different gene regions that have been linked to coronary artery disease, and this is a very unbiased assessment. Now we talked about the modifiable risk factors. Many of them do also show association with modifiable risk factors, but it turns out about half of them do not. It's arguable that not all of them may be fully explained by the modifiable risk factor associations, but many of them we actually don't have a great hypothesis today in 2025, because we obviously appropriately focus on modifiable risk factors, but we've not had the tools yet to really do an unbiased scan to start our hypotheses now directly in the target organism. And there are lots of challenges to unpack the mechanisms of each of these. We think about this. Many folks in the field think about this. What has emerged now from the totality of the data, what one can do without understanding the mechanisms of directly that, what is responsible for that genotype-phenotype correlation, to take that all together as a predictor, very similar to the clinical 10-year risk score, where you have age, sex, diabetes, cholesterol values, smoking, and then you have a numeric value. This can have, several thousands of SNPs and oftentimes bigger, where then you say, okay, at the population level, these are the SNPs that are enriched for cases. Now for the patient in front of me who has a family history of cardiovascular disease, are they also enriched for the alleles that are present in cases relative to controls? And more contemporary methods will look at the relative enrichment for cases and controls, and so then we can weight each of those SNPs because their prognostic relevance is variable. Each of those individually are very, very poor predictors. However, together, they can be more meaningful predictors. And as I said, it's a trait of interest. We focus on coronary artery disease, but now, our data sets and biobanks can analyze, pretty much any condition, including the other common cardiovascular disease conditions. All of them have a heritable basis because obviously, you need the genes to manifest yourself and the genes to manifest disease. And it turns out that genetic variation does influence some of the variability that you see in the population. And what's interesting, DNA sequence variation is pretty similar between two random individuals. It's about 99.4% similar. So we're looking at a very small margin relative to the human genome of variation that does seem to influence in sizable fraction, it really depends on the disease, influence in the variation that we see for whether people get disease. Now, like familial hypercholesterolemia, interestingly, the polygenic basis, so the multiple different additive increments of common genetic variation that are influencing many different pathways to get to the risk factors, also seem to influence risk for coronary artery disease beyond just the measured risk factors. Now, what we do today, when you do these 10-year risk calculators or when you go to the doctor's office, you're pretty much focused on that value in clinic at that time. What we think this is doing, and the reason why the genetic basis is accentuating that risk, is that it likely is integrating information from the prior visits, from the unmeasured values as well too. That's the hypothesis for familial hypercholesterolemia as well. And interestingly, obviously, as I said, the prevalence of risk factors is lower earlier in life. However, these polygenic risk scores are basically the same. So they can also predict the onset of risk factors as well. There is this concept of primordial prevention and thinking about how do you prevent the onset of cardiovascular disease risk factors. That's largely in the space of lifestyle optimization as well. But really now we can start to better pinpoint with maybe some degree of precision of who might then develop these modifiable risk factors. But really in the end, what we want to do, especially as cardiologists, is say, who is going to get disease? Who is going to get coronary artery disease? And the tools that we have are increasingly good and increasingly being refined. But we do recognize there are multiple different factors that influence the likelihood of disease. And so the polygenic risk scores have been continually evolving. This is an earlier iteration of a polygenic risk score, really simply looking at the question of family history. Is it just getting the family history information and not actually telling us more? Now, the distributions are different among individuals who have a family history. It's higher on average. The score is higher on average for folks who have a family history for cardiovascular disease. But in those with and without, it is actually predictive in both scenarios. It is slightly better among those who have a family history. And I think the important thing to realize, and a challenge for the field, is there isn't yet a canonical way to represent a polygenic risk score. There are many different methods, and the field is evolving because the data sets are getting larger and they're representing more individuals. So the scores are getting better, but oftentimes the same hypotheses that have been assessed in the literature need to be reassessed, and the effect estimates continually change. And it's a problem because, if you come here to get your blood lipids drawn, if you come to my hospital at MGH and get the blood lipids drawn, if it's the same sample, you'll probably get the same thing. Now, there are different, because as I said, there are different ways to calculate polygenic risk scores, largely because the training data is changing and there's some aspect of tuning. You may not get the exact same thing. The performance at the population level is continually increasing. Anyway, we'll come back to that concept. So when people talk about predicting risk, a metric that they often focus on is the C statistic, for better or for worse. It is a measure that basically says, those who develop disease, are they scoring high on your test? And if those who don't develop disease, are they scoring low on your test? And 0.5 is basically a flip of a coin. These are the risk factors that we use in our 10-year risk score, because I think it's always important to compare to the things that we use today for benchmarking. And you can see, these are better than 0.5. These are the reasons why we use them today. And this is one such score that, is basically on par, maybe a little bit better. But obviously, we look at the totality of the risk factors together, and we're still getting more information at the population level. So we said, that was in the UK Biobank. We said, okay, let's look at our biobanks where we have, great clinicians, right? So this is within ours at MGH, at the University of Pennsylvania, and Mount Sinai. All of them have biobanks, all similarly designed and a similar size. This is, an earlier iteration. We calculated the same polygenic risk scores and made a couple observations. One, surprisingly, despite each of these health entities being in different geographic areas, taking care of different types of patients, the risk signal, this is the effect estimate per standard deviation of this continuous biomarker, was the same, nearly identical across each of these, suggesting that despite, contemporary care where we take good family histories and maybe we have, highly attuned clinicians, there is still some risk signal there despite what we're doing. Now, if you look at, readily available risk factors, including the distribution of the predicted risk based on conventional risk factors, you basically can't identify these individuals. Individuals with FH, as I said, you have a pretty good screening test with blood lipids. Those who have a high polygenic risk score, they have a very mild increase in LDL cholesterol, almost imperceptible, about 5 milligrams per deciliter. That's like, 0.1 standard deviation units. So nothing that you can reliably detect. And then if you look at the bins of individual, risk categories based on 10-year risk prediction, those who would be identified as being eligible for lipid-lowering therapy versus not, that distribution is basically the same whether you have high or low polygenic risk. So we said, in 3 different health systems, we have a risk signal. These folks at higher risk for getting disease, pretty similarly so, but we are not identifying them today with our current metric. And this was really motivated, so this was done, a few years ago. This was motivated actually after the last guidelines, this was in 2018, that introduced this new concept of a risk enhancing factor. This is saying we have the traditional risk factors, but there are other things that we know that if patients have can further upclassify risk. And it's a pretty loose evidence basis of like what actually made it onto that list, but roughly is 1.5 to 2-fold increased risk. Not really good for de-risking, but saying if there is some clinical equipoise and you're not sure what to do, if they have additional risk factors, okay, maybe you should be a little bit more aggressive. So we said, this is actually a pretty similar evidence basis depending on what you have your cutoff for high polygenic risk. And then we went back and we said, maybe doctors are pretty aggressive, everyone's probably going to get treated anyway. So you subset that group, at least within this very curated heuristic that is consistent, is at least aligned with the evidence. There's about 4% of individuals who don't have cardiovascular disease. If you use this metric, we said you'll look at estimated 10-year risk and say, okay, there's already some equipoise, but they have this additional factor, you would recommend statins. Many of them are treated, but a subset are not. So one of the major challenges to traverse into clinical care has been differences in performance in different groups of individuals defined by genetic ancestry. And this is actually now a classic figure by one of my colleagues in Boston. If you look at the rise of the different populations of individuals in human genetic data, it has largely been Eurocentric data, cohorts like the Framingham Heart Study and the UK Biobank, and this is relative to the world's breakdown of the different ancestry groups. And this is kind of not unique to any risk predictor. Your risk predictor will always predict disease or a condition best in where the training data is best represented. So these polygenic risk scores, this is a figure looking at the distributions of performance of polygenic risk scores for multiple different traits relative to individuals of European ancestry. And they all basically decay from European ancestry. And again, that's not because Europeans have more genetic disease. It's just a function of what is in the data. And there has been a recognition of this for at least 10 years in the field. And so there has been a concerted effort to increase the groups of individuals that are in these human genetic data sets, because obviously we take care of lots of different patients. The US's response is the All of Us program. This was kind of in 2015 at President Obama's State of the Union address when he announced the Precision Medicine Initiative that ultimately morphed and was conceptualized in the new Framingham for the US. That's the All of Us research program. That is nearly at its target goal of 1 million, maybe about 800,000, and so far as whole genome sequencing, and about 300,000 individuals across each of the states. And we very recently said, okay, if it's supposed to reflect all the Americans, does it actually reflect all the Americans? We compared to the US Census. And it does a pretty good job of individuals who are African American, European American, but there's a depletion of individuals who are Asian American, East Asian, and South Asian. And this was important because, one of my motivations was, especially as I was increasingly recognized that this cardiovascular disease, diabetes, was increasingly common in South Asians and we should study it. It's clear that we just don't have the research infrastructure to study it. And there are many different reasons why some groups of individuals participate in research and others don't. So we designed a study, and all of us like study, actually very much harmonized with this research study, to then go out into the community and collect samples for individuals who are South Asian ancestry. So please feel free to share with friends. Now, the scores, as I was hinting at, are continually changing and in general, continually improving. I say in general because there isn't yet been a plateau for coronary artery disease. There's probably been more of a plateau for BMI, for example. And this is a comparison of two scores. This is a score that we had recently, and this is a prior score from our group. And this is a metric that is aimed at trying to make a parallel with monogenic risk. As I said, familial hypercholesterolemia, 1 in 300 individuals, about a 3-fold risk for coronary artery disease. That was compared to non-carriers. Now, if you take individuals, if you're trying to identify what is that cutoff for a 3-fold risk compared to the average score for a polygenic risk score, and the older score is 8% of individuals, and the newer score, again, the genotypes are exactly the same. We just can interpret them better. It's now actually up to 1 in 5 that has an FH-like relative risk that can be explained from a polygenic model. But we can't readily detect by really high LDL cholesterol. Because the training data is getting better, the relative effects are actually increasingly similar across populations. A uniform and consistent observation is those of African ancestry, despite actually having probably the second most in training data, continually have lower relative risks for multiple different traits, including for coronary artery disease. That's important information if you're going to represent this back to patients so that it's more accurately reflected back to patients. But nobody has a great explanation for why there is a lower relative risk. It could be due to competing risk, but it suggests there are key modifier effects as well, too. Now, there have been prescient studies of actually already prospective studies of returning this back to patients. This is a study done at the Mayo Clinic using an older polygenic risk score, but really conceiving of this notion of individuals being at intermediate risk and then separately giving them a polygenic risk score. And there's a couple things that you observe here. Basically, when you tell patients their risk, regardless of their risk for cardiovascular disease, LDL cholesterol decreases. This is a mix of both pharmacologic and non-pharmacologic things, so really shows the importance of talking about this in the individual clinical encounter. But those who are specifically told that they had high genetic risk, that led to even lower LDL cholesterol. And you could say that's great, it's motivational. If you lowered LDL cholesterol further for anybody, including the lower risk individuals, maybe they'd benefit as well. Now there's a surprising observation that individuals who have high genetic risk seem to benefit more from lipid-lowering therapy. And I'll walk you through this because this is not exactly how they were designed. But we went to clinical trials that were completed for statins. Statin versus placebo is shown on aggregate to have a lower risk for cardiovascular disease by about 0.7 to 0.8. And today, if you do subgroup analyses, or what has been done subgroup analyses, if you look at other enriching factors for why we prescribe statins like smoking, diabetes, hypertension, all those individuals will have the same relative risk. The reason why we're prescribing to those individuals is because they have a higher predicted risk. They have a higher absolute risk. So with the higher absolute risk, the same relative risk reduction, you have a greater absolute risk reduction just because they're at higher predicted risk. Now, for those who have a high polygenic risk score, you have a higher predicted risk, so higher absolute risk. Now, interestingly, there's a greater relative risk reduction for cardiovascular disease. You're lowering LDL cholesterol exactly the same. What that does is that further magnifies the absolute risk reduction, and that comes back to this number needed to treat argument, which is basically the inverse of the absolute risk reduction. Most ideally, if you're going to recommend for a large group of individuals to be treated, particularly asymptomatic individuals, you want to increase the likelihood that somebody is going to benefit. Because, in the office, especially in preventive cardiology, those patients, they're feeling fine. The goal is not to make them feel any better. You want them to not get disease. But if you're going to make a recommendation for a medicine, that's basically a daily pill for the rest of their lives. And many of these folks are younger. So there is the crystal ball and refining the crystal ball, but that is a means towards at least making a confident recommendation where you say, okay, you are more likely to benefit from that medicine. And this actually is a new tool that specifically helps with that conversation. Now, this interestingly is not unique to statins. That's why I generalized to lipid-lowering therapies. This has been done for the PCSK9 inhibitor trials as well, too. For both of those that were shown to lower cardiovascular disease risk, those who had high genetic risk actually benefited the most from those medicines. Now, we have done some work among individuals who then develop disease. And part of this has been motivated by specialists, because a lot of people will refer to me patients, not just at risk, but those who have developed premature cardiovascular disease, and wondering if there is a genetic basis and a genetic reason why they develop cardiovascular disease. So one, want to understand, okay, sure, they probably have a high polygenic basis, but would that have any prognostic information? Now, in the UK Biobank, we took all the individuals who came in having had coronary artery disease and then looked at their future risks. And the nice thing about the UK Biobank is that they're profiled for lots of different things. Clinical risk factors, obviously genetic factors, but some of the other socioeconomic factors. And you kind of put that all into the model. And if you look at explanatory variables, surprisingly, the top explanatory variable was the risk factor that was there before, their polygenic risk score. And you might say, hey, look, this is totally suspect because LDL cholesterol is down there. And that's because, at least what I think, is that when patients develop cardiovascular disease, we are very attuned to treating their risk factors. We are constraining them to normal ranges. We are bringing them normal and often below normal, especially when it comes to LDL cholesterol. So at the population level, those don't become meaningful predictors for recurrent risk. It's the things that you're not treating. We're obviously not directly treating the polygenic risk score. So it turns out that continues to be a persistent signal for recurrent events. And this is a collaboration with folks in Italy for that specific scenario of folks who have premature early onset myocardial infarction. It does predict the likelihood of a second, third, and fourth. Now, where I think the real opportunity and the real impact is going to be is early in life. Because I basically started this by saying atherosclerosis starts early. We can't identify those individuals early enough because we're basically waiting for 10-year risk. So we're basically waiting until middle age. And there is a general awareness that that's an important problem and that people have already started to think about these scores in younger individuals. And the tools that we've been using, again, since the inception of the guidelines was to focus on individuals with severe hypercholesterolemia. And we should continue to do that. And obviously, that is still a major issue, is still undertreated. But will not fully solve the problem of identifying all those individuals who are then likely to get coronary artery disease, and particularly those who are going to get premature disease. As I said, if you use a very similar heuristic, and again, a lot of this stuff is motivated by saying, what do clinicians do today? What do the guidelines do today? What's the evidence basis? Risk prediction, 10-year risk prediction, there's no randomized control trial based on that. It's hard to do. There are many different challenges. One can argue on both sides whether you should. Screening for severe hypercholesterolemia, when it was recommended, there's no prospective study that says that has reduced outcomes. Actually, that's a reason why the U.S. Services Preventive Services Task Force has not recommended pediatric screening, even though the American Academy of Pediatrics does. It makes general sense. Anyway, using that same heuristic of a predictor and likelihood of benefit, you may actually, we would actually identify many, many more additional individuals if you use this test earlier in life. And obviously, it's not an either-or scenario. We would obviously want to incorporate all this information together because, for somebody who develops a heart attack, it's usually not one thing. It likely is a myriad of multiple different things of why they ultimately develop disease. Here is our ability to capture events at different ages. This is in the UK Biobank. The blue and the green together are basically our ability to capture the events based on clinical risk prediction. And then the pink and then the overlap with the green is based on using genetic information. So you can see complementary, like we can find the events later in life based on clinical risk prediction. We can find the events earlier in life based on using genetic information. And so once you put that information together, this is that C statistic. So if you just use what we did today, its performance is not great for premature events, and then it gets better if you're lucky to have an event later on. But if you add this additional information, you get much more stability in performance across the life course. Now, we do in the clinic use a lot of imaging. Imaging is great for predicting cardiovascular disease. Imaging, the utility of imaging, CT-based imaging for predicting risk, has a very low diagnostic yield in younger individuals. But when you look at individuals who are like in their 40s and 50s, and you look at what do the subclinical plaque features look like for those who have high polygenic risk, those who have high polygenic risk are more likely to have asymptomatic blockages and are also likely to have higher features of plaque instability based on CT angiography. And if you look at this serially, they're also more likely to have plaque progression. Now there is many people say, okay, we should modify their risk. Maybe that's the thing that we should do. Maybe we shouldn't do lipid-lowering therapy. I don't think that's a great argument because we kind of have to do that for everybody, right? Most ideally, we would optimize everybody's lifestyle. That's very easy to say. To actually operationally do that in the clinic is very challenging because there are many individual issues why people do the things that they're doing and not doing the things that they're doing. And many of the things are in their control. And in my experience, most of it is also outside of their control. However, if you look in the observational setting, as we said, there are many different reasons why people develop cardiovascular disease. Those who have high polygenic risk, who do have a more favorable lifestyle, do have lower cardiovascular disease risk. But there's still an important residual risk depending on what bin you're at. Now, this information can be motivational. This is from a research study in Finland where they disclosed about 6,000 individuals clinical risk, genetic risk. Many of the people felt like the genetics might tell them something important about their health, very similar, maybe even higher than the direct-to-consumer genetic testing survey. But if you look at the degree of their risk and then the likelihood of improving their lifestyle when they then surveyed these individuals in follow-up, both separately, the extent of clinical risk and the extent of genetic risk were important motivational factors in optimizing their lifestyle. Again, I said agency is one small part of it, but again, can be a complementary tool to an overall agenda of improving cardiovascular health. But what I'm excited about is really refining and improving these predictors across the life course. This is pretty early work, and this is it capturing much more clinical information beyond the cardiovascular disease risk factors. Somebody who is going to get, somebody who has breast cancer or rheumatoid arthritis, that information likely influences the likelihood that they get coronary artery disease. The various different medicines in the social context also likely influences the likelihood of getting them a wide variety of disease on the background of a starting trajectory of polygenic risk. So we've developed this model. This is using the general practitioner health records, those individual level records that are available in the UK Biobank, and then actually externally validated within our health system, the ability to predict about 300 different conditions. And again, if you look at that C statistic, increasingly using both the genetics and the totality of health information will be very important for both predicting coronary artery disease and others, really as a means to moving the needle earlier for those who are at particularly high risk. As I said early on, one can, with the same tools, calculate polygenic risk for anything. And because most of the literature is obviously focused on a single disease area, but if you just take the common cardiovascular disease conditions, this is in our MGB biobank, there's a pretty high fraction of individuals, this is not in the Mendelian space, who have a high polygenic burden for future risk for cardiovascular disease. And obviously these have familial implications, as you might expect, tight correlation among twins and then expected correlations with other family members, very similar to monogenic testing. So really it's based on all that we developed a test. I mean, some of the other things I didn't really cover, but as I said, because of the varying performance of different scores, it is really important for us, for trusts and clinicians, to understand how the scores are being developed. There are commercial assays out there that we don't really know how the scores are being put together, and much of that led to us developing this. And then, I have a separate hat in the entity that does in-house genetic testing. So we're able to pull all this together. The methods and the tools are all transparent and open, and the code is all open source. But as I said, today, clinicians anywhere can order this test. Okay, so, in summary, polygenic risk scoring, I think at its root cause, its root clinical opportunity is really to help us interpret the family history in young adults, really using it in concert, obviously, with clinical risk factors, but really help us identify those who are at particularly high risk earlier in life. That might involve medicines, that might involve additional testing, like CT-based testing. And I would argue that, if you can better refine who is at risk earlier in life, your preventive therapies are more likely to work, particularly for premature events and then for lifelong events. And that's supported by, other evidence that we didn't talk about. And, a challenge, but also an exciting challenge, is that the scores are changing. So how do we as a field, if we're going to represent this back to patients, how do we acknowledge the dynamic nature? How do we feed information back? And then also, there's no clinical test today that you order and a lab is going to synthesize the totality of the EHR and then present that information back. And we don't want to represent single risk factors back, because that would not be an appropriate representation of risk, because it can get misconstrued, because people have a lot of things in their mind of what genetics is. So we want to evolve to incorporate these additional factors back. And then importantly, there is an opportunity for multiple diseases, and I think it's still important for us to understand how it might be used. So these are all the people that do the work, and I get to stand up here and talk about the work. And Shri is actually one of the former, or actually active member in the lab now, medical student here at Case, so I'll call her out. But thank you. I'll open it up for questions.
Going back to one of your early slides, when you talked about familial hypercholesterolemia, you noted that for a given level of elevation of lipids, serum lipids, that familial patients with FH were at greater risk.
Yeah.
Is that due to genetics, environment, or other clinical risk factors, or both?
Yeah, well, we think that it's driven by a cumulative burden of LDL cholesterol that precedes that single time point. Like when we're looking at the lipids, when we go in the office, we're basically focusing on that one. And obviously, we'll look back, but there isn't a quantitative heuristic that brings all that information together. The genetic basis of it, we did this in the paper as well, in one of the studies, in the ERIC study, where you can see those individuals, when you go back, they obviously have consistently higher. So we think it's largely responsible. Largely, I say, because if you look at an equivalent polygenic load for high LDL cholesterol similar to FH, it doesn't accentuate risk as much. And I don't know why, but that suggests that there might be other things.
For people with FH, if you exclude the FH allele and do a polygenic risk score on the rest of their genome, what kind of variation do you get?
Yeah, so important terminology. FH is used loosely. It's technically a genetic diagnosis, but it's often used, with severe hypercholesterolemia and a family history. Among those groups of individuals, there's actually only about 1 in 50 who have an FH mutation. And depending on this, the last time we looked at this was maybe 6-7 years ago. At that time, it was about 10% that had a high polygenic score. We now actually we have this information in the panel. One of the reasons that we have it is not necessarily specifically to look at severe hypercholesterolemia, but in feedback from clinicians, they also do want to understand the extent to which, if somebody has a high polygenic risk score for coronary artery disease, is it a high polygenic load for a specific risk factor? Interestingly, that's actually pretty uncommon, because it's integrating lots of different risk factors, often not being driven by a genetic basis of one.
So if you intervene early, you might actually see that come down.
Yeah, and as I said, we didn't talk about this, but probably there's at least 2 key lines of evidence that suggest that treating early is beneficial. So one, if you look at the randomized controlled trials for statins, I threw that number out a bunch, about 0.75 for a hazard ratio. That's for the end for the follow-up. But actually, if you look at each individual year that the participants are in the trial, that hazard ratio is continually magnifying. It's not a proportional hazards. It is cumulative for the number of years they have lower LDL cholesterol. And then if you go back and you look at, PCSK9 loss of function mutations, about a similar reduction in LDL cholesterol, that hazard ratio is about 0.1, 0.15, suggesting that if you make that perturbation at birth, which we're not necessarily arguing for, again, it suggests a cumulative effect. And that's why it's important to identify risk early if we can.
I'll follow up on that, and then one other question. So if you see someone in your 20s in your clinic with a top 1 percentile of polygenic risk score, and even though at that age they don't have high LDL, why not recommend a statin to lower their LDL burden over their lifetime? Would that be something you would consider?
I would consider, but there are a lot of unknowns, right? I think the challenge is, it depends on who comes into the clinic. I will say that the patients who are seeing me are not necessarily the exact same patients who are out there in the population who are in their 20s. Most 20-year-olds are not super excited about taking a medicine. I think if there was also a family history, that would be a compelling argument. If there was a mild perturbation in the LDL cholesterol, that would also be a little bit more of a compelling argument. There's pretty good data that suggests that the optimal LDL cholesterol is probably not the average LDL cholesterol in the population, which is about 100. The optimal is probably closer to 40s to 50s. Okay, so even if somebody doesn't have a high LDL cholesterol, in general, you could probably still optimize their risk. But I do think for it to be like a good use case for that patient, you probably need additional factors. But let's say they're not on board with it. That's not necessarily incorrect. This is why they, continually do follow-up. If the LDL cholesterol is now going up, the context is very different to maybe a 30-year-old who is coming in follow-up who has mild hypercholesterolemia. Not to add too much information, but this interaction for lipid-lowering therapy and greater relative risk reduction actually seems to work in the reverse. Like if you have mild to moderate hypercholesterolemia and you have a high polygenic risk score, that risk actually is not just additive, it is multiplicative, and it gets you closer to that severe hypercholesterolemia risk. There's a paper that's just submitted, basically defining different thresholds for a high polygenic risk score based on your LDL cholesterol.
And the other question is about the ancestry. So the PRS performs very poorly, or much more poorly in the African ancestry population. Is that because the training populations are still European, and you're throwing in a lot of SNPs in Europeans that are not in LD with anything in the African population? But if you train it, now we don't even have SNPs designed to get good genotypes of the common variants in African ancestry potentially. And if we use those SNP arrays, maybe we would we could develop a good PRS for African ancestry. Do you think that's the case?
Yeah, this is a really important issue. Now, I'll say that the scores are predictive and basically in all the populations, but they're predictive variably so. And this is the case for basically any risk factor as well. They have different distributions in different populations. They have different fold effects. But to be honest, today in medicine, we just kind of use the same cutoffs and the same thresholds. But I think I'll say that we're imparting some more nuance by saying that the relative effects are different. Now, the reason why, as I said, I don't have a clear reason for it. Some of it could be, some people have theorized that the canonical human genome reference may have intrinsic issues that it might lead to poor mappability from African individuals. So maybe in reality, we have poor coverage, and Scott being an expert in that area. I do think that there are probably other factors that we don't know that do modify risk. And there has been a lot of effort and interest in trying to study this. Now, everybody had hoped that we could study this in the All of Us research program because they actually ask lots of contextual factors across the US. The problem is the surveys have so much missingness. Like maybe there's like 10% that have a complete survey. And obviously the completeness of the survey and what you answer is an important factor for why you get disease. So, it is kind of disappointing, but this is information that still needs to be collected and understood. And it's also important because, in the US, African Americans have a wide variety of African and European admixture. And that likely influences their particular fold risk. And we've developed methods to better represent across admixture. That hasn't yet traversed in the clinical space, but maybe will.
So I'm still struggling on how to practically incorporate the PRS and the risk evaluation. If you have a multivariate linear regression model and with all the modifiable risk factors and you include your PRI score, what percent of the variation does that contribute to the risk?
Yeah.
I imagine that must be quite small. So then this leads to the next question. Why would you do a genetic testing before, even at the time you do all the other evaluations, and use a traditional approach? We didn't even talk about the microbial, microbiome there. But so what percent of the variation is accounted for by the PRS?
Yeah, actually, I didn't show this paper, but we did this exact experiment that was published earlier this year. It's very similar to that recurrent risk study. However, the top predictor is hypertension. The number 2 is a polygenic risk score. In general, they still represent a minority of the variation. But if you rank the different things that we, obviously all the things that we use are much lower. And I hesitate to do comparisons because what is not intended to say let's use one in lieu of another. But if people are benchmarking things, I think it's really important to compare it to the things that we use today. But I do acknowledge there are other things that are going to influence risk, the extent to which the microbiome does. We don't know, and partly because it's not been assessed in an unbiased manner in large populations of individuals. So we don't know if that's going to be really high. We don't know if that's going to be really low. Those are, I think, intriguing hypotheses. But the fact that that missing gap is still really high suggests that we need to look at other things, beyond genetics and other factors.
Thank you, Dr. Natarajan. On slide 37, I think you had covariates ranked by their contribution to the prediction model. Is that how I would interpret that?
Yeah, this is for recurrent risk.
Yeah, so I've just been thinking about causality lately. So correct me if I'm just completely off on this. But a lot of times when we think about causality, we think of predictive causality. There was an economist in the 50s that really popularized that idea that an economic model is completely graded on its ability to predict. But the more I think about it, it's not clear to me that that's the most relevant thing for clinicians. Like clinicians would think like maybe interventional causality. And it's not exactly clear to me that if you intervene with HDL cholesterol, that's more important than intervening with LDL cholesterol.
That's totally accurate because actually there are multiple medicines that have been designed that raise HDL cholesterol that don't improve risk for cardiovascular disease. It is still, despite all of that, because those trials go back to 2007, that in 2025 is still in risk prediction equations because it still is capturing risk to some degree. And what this argument is not saying, okay, we need to intrinsically change these things. I'm not arguing for CRISPR across a million different parts of the genome. Obviously that would not make sense. However, this case is very different than I think where the key opportunity is, which is preventing the first onset. The fact that it's predicting recurrent events, no clinician would change what they do. However, there are millions of medicines that are being developed, lots of medicines that we would then potentially recommend for patients, and the guidelines have started imparting some stratification for recurrent events because we acknowledge that maybe not everybody would benefit for throwing the kitchen sink at them. And that stratification is based on actually these factors that have not yet included genetic factors. It's been basically these factors, if they have an accumulation of these factors, these may not be the causal things that you focus on with your particular intervention. But if you're going to step up intervention, focus on these folks who have an accumulation of factors that have recurrent events. It's that concept of having high absolute risk, but the relative risk being the same. That, again, coming back to the primary prevention, preventing the first event, the fact that you might actually have a greater relative risk reduction, that's actually pretty compelling for primary prevention and really maximizing your therapy for asymptomatic individuals. But yeah, great, astute question.
So great, thank you for a wonderful talk. I want to encourage everyone who still had questions, or if you didn't, we have a reception afterwards with drinks and food and a chance to talk to Pradeep again. And just join me one more time to give him a hand for the presentation.